There are two types of immunity, or at least when we consider the immune system, the first thing we do is to break it into our innate responses. This includes anatomical barriers, antimicrobial chemicals, and a number of white blood cells such as neutrophils, eosinophils, basophils, monocytes, etc. But then there is another type of immunity known as adaptive immunity or acquired immunity. In the innate immunity video, I asked, and I would ask my students, were you vaccinated? Where were you born? Where did you go on vacation? Oh, wait, I don't care. And in innate immunity, we don't, because innate immunity does not change over time. You still have acid in your stomach and salt in your sweat. You still have neutrophils, which phagocytize bacteria, or eosinophils, which attack worms, regardless of where you were born or where you went on vacation or whether or not you were vaccinated. However, that's not enough. So if your antimicrobial chemicals and anatomical barriers are your first line of defense, those white blood cells I just mentioned are your second line of defense, that's not enough. It might be if you were a bug, right? A typical bug life cycle might be you hatch from an egg, say in April or May, and then by September you're dead. And if that's your life cycle, odds are you can make it your entire life with, say, 2 lines of defense, all right, without an infection getting so bad that it takes your life. However, we hope to live decades. We don't become, reproductively capable until, second decade, give or take. And so obviously, we vertebrates, we look at life differently. We don't measure it in months. And so therefore, 2 lines of defense is not enough. And so therefore, there is a third line of immune defense that only the vertebrates have, involving lymphocytes. So while there were all of those white blood cells that can differentiate in the bone marrow that were discussed in the innate immune system, the adaptive immune system revolves around one type of leukocyte known as the lymphocytes. It is because of these lymphocytes that we can live years as opposed to more on the line of months. This is where our adaptive or acquired immunity comes from. Now, the number of lymphocytes in our bodies is enormous. They would equal more or less the weight of the brain if they were all in one place. And they are incredibly complex. So obviously the brain and all of the neurons are complex. If you were looking for the second part of your body, which is most complex after, say, neurons in the brain, I don't know anyone who would argue that it's your lymphocytes and the immunity that they provide. So this is so complex that now we have to break this into 2 parts. All of these lymphocytes, they begin in the bone marrow. That's where they are formed. Some finish their development in the bone marrow. Others go to the thymus. The ones that begin, that finish their development in the bone marrow, they produce antibodies, and we call them B cells. I know it's easy to remember that, they were made in the bone marrow, but actually they were first discovered in the bursa of chickens, and it's just dumb luck that begins with a B. The T lymphocytes, which will be the topic of the next video, they mature in the thymus. They have receptors which are hypervariable, just like the antibodies, but they're not secreted, so T cell receptors are different. So we split the adaptive or acquired immunity into 2 parts. The humoral immunity, which is primarily involving the B lymphocytes, and then the cell-mediated immunity, which primarily involves the T lymphocytes. This video will go over the humoral immunity that the B lymphocytes are the major players in, and the next video for the T lymphocytes. So B lymphocytes, they make antibodies. What's an antibody? Well, it's a very complex protein. Well, so that's good. Lots of proteins are complex. But this one is special. The jawed vertebrates, they evolved a mechanism, and it was actually maybe even after an infection. So it might have been a virus or a transposon which split up a gene that, so like an infection actually made the jawed vertebrates better at fighting infection. So that's neat with the RAG recombination. But in any case, Our antibody genes, we could really call them super genes. So if you look here, here are all of these pieces. A normal gene says, makes parts of a protein that you mesh together, and here's the protein. But here, so there's all of these sequences for, let's say, a blue section, a red section, a green section, a purple section. And each B lymphocyte is going to make one kind of antibody. So let's say lymphocyte number one, B lymphocyte number one says, all right, let's make an antibody. So it chooses that B section, that red section, that green section, that purple section, and it makes an antibody that looks like this. And it just turns out that's very good at binding to streptococcus. Another B lymphocyte, once again, each will only make one kind of antibody, takes a different B section, a different red section, a different green section, and it then makes an antibody which is very good at binding staphylococcus. And another one will choose a different order and then make an antibody which is very good at binding bacillus. So with this one gene, we're not making one protein. And in fact, we can make billions of different kinds of antibody. Now, our genome only has 26,000 genes, give or take. Being able to make billions of different proteins, that's really significant. And it's very significant that we can make antibodies against, say, Staphylococcus aureus or Streptococcus pyogenes or Bacillus anthracis, et cetera. And just an analogy, and we'll obviously get to this in this video, but neutrophils, they can recognize bacteria, so why are we making a big deal about this? So yes, neutrophils can touch something and say, ah, you're a bacterium, maybe I should eat you. So neutrophils can recognize bacteria and attack. This B lymphocyte is going to recognize one. All right, so Streptococcus, or this species of Staphylococcus, or this strain. These antibodies are very, very specific. As a result, if there comes a day in your life where you have a serious infection caused by not bacteria in general, but one bacterium, Streptococcus, these are now white blood cells, which will go after that one and only that one. All right, so if you think in a crime scenario, yes, you want, police in your area looking for, any old person who might rob a store or any old person who might, be driving drunk or whatever. But let's say there's a very serious crime being perpetrated by one specific individual. If that's really serious, you want, officers who are going to focus on that one, individual or gang or whatever and focus in on them. And so we can create billions of different antibody combinations. T cell receptors are kind of like this as well. They can generate millions of different combinations. So there's lots of bacteria in the world. And yes, neutrophils can identify bacteria. But what lets us live so long is that we can provide a very specific defense against one kind of bacteria. So this B lymphocyte is going to go after the red bacteria with its red antibodies. All right. But a different B lymphocyte is going to take different pieces of that gene, assemble them differently, and then go after pink bacteria. Another one goes after, say, green bacteria, et cetera. And so antibodies have this incredible diversity. That's the key to humoral immunity. This is to represent a T cell receptor. It's not Y-shaped. T cell receptors can have millions of different combinations. And so our ability to create billions of different kinds of antibodies, that is a key aspect. Now, these Y-shaped proteins, it's the top part which is actually going to interact with the infectious agent. And this bottom part, the base, can actually be switched out. So I'm going to change the color of these sections here. Now, changing the base doesn't affect the bacteria which you can bind to. So as long as this top part is there to bind to bacteria, you can change the base and that's fine. But these, the switching of these bases, that is actually important. So the constant heavy chain can be switched. Let's say we have this blue section. Well, in this animation, we'll call the blue section IgM. So if an antibody looks like this, it's what we call IgM. Ig stands for immunoglobulin. This is what B cells have on their cell surfaces, receptors. And if it's secreted in a group of 5 known as a pentamer. And we'll get to why that's important in a second. So if this antibody has that base for IgM, it's a receptor on B cells, it can be secreted in a group of 5. However, if we take the same antibody reaction and now we just put on a different constant heavy chain, now it becomes IgG. This is secreted into blood plasma, right? And so if we want there to be antibodies in plasma looking for microbes, then we make single IgG molecules. And then we can do that just by switching the base. If we put on this yellow base, that then gives us the antibody IgA. These are secreted from the body. They're not in the blood, but nevertheless, they are lining the small intestine. They're in breast milk. They are in body's secretions. Now, we probably don't talk about IgA as much, it isn't mentioned as much. However, just because there are so many microbes in the digestive tract, the amount of IgA which is made actually exceeds all of the other types of antibodies which are made in volume. So we make a lot of IgA. IgE is of interest, and we'll get to this at the end, because if I make IgG in response to, say, grass pollen or peanut butter, molecules or whatever, then I am not allergic to those things. If, however, I make IgE, all right, remember it has the same binding capability up here, I'm just switching the base. I make IgE, that now means I'm allergic to grass pollen or allergic to peanut butter or bee stings or whatever. So IgE, because it binds the mast cells, as we'll see, it is responsible for allergies. Okay. So those a little background into antibodies. Now let's talk about how they work. So here we have a B lymphocyte. Notice it's making a yellow antibody, not a yellow and a red one, or a yellow, a red, and a green. Even though there are billions of possible antibodies, this B lymphocyte only makes one kind. That's why it will be able to mount a very specific offense against one specific bacteria. And so after it binds this bacteria, a couple things can happen. It divides, all right? Now, you'd think that, oh, doesn't it eat bacteria or doesn't it? No, 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 no. It divides, all right? So it's going to attack bacteria differently than a neutrophil that's going to, like do phagocytosis and go about it that way. It divides to make a memory cell and a plasma cell, right? Now, the memory cell is going to be around for later, while the plasma cell makes the antibodies which are going to combat the microbe. All right, so why this memory cell? Well, that original B lymphocyte, if you were to ask how many lymphocytes in the body are making those yellow antibodies able to react against that specific microbe, the answer is we don't know. I'm just going to make up a number. Let's say, I don't know, 3, 5. I mean, if you have billions of different kinds of antibodies, you can't have too many of any one, all right? Because, you wouldn't have enough cells in your body. So even though we can make antibodies against a specific microbe, we don't have, an endless number of those B lymphocytes. It's a smaller number. So where are they in my body? I don't know. Let's say one's in a lymph node, or one's in my spleen. One's in a Peyer's patch in my intestine, one's in a tonsil, all right? So I've got a couple and they're dispersed around the body. If an infection starts and this microbe starts to spread, my humoral response depends on one of those bacteria contacting one of those B lymphocytes. But if they're spread all over the place, it will happen. But the problem is it might happen later. And by then the infection might be, far gone enough that actually, even though I'm reacting against the infection, my life's in danger, all right? Because the bacteria spread to that degree. So Next time, if I survive this infection, now that the body has recognized, aha, yellow antibodies, that's what we want to make. You know, there are microbes that bind to this that are in our environment. We want to be on the lookout for these. I won't have 5, you know, cells capable. I'll have hundreds. And so the next time, if there is a next time, my reaction will be so much better. All right, so this time I'll have what's called a primary immune response. And it'll be okay, I hope, to survive. Not super overly impressive as immune responses go. But now let's say later, I now get infected with the same microbe. Now I've got these memory cells, all right, which will start this reaction faster and then start a secondary immune response, which will be so much bigger. So if you measure the antibodies which someone is making against a microbe in their primary immune response the first time they come in contact with it, and a secondary immune response, the secondary response is so much bigger. The first time around, my life might have been in danger. I really might have been struggling to get through this infection. But the second time, I might not even notice that I'm sick. All right, my immune system might be that good at going after this microbe. So here's the point of adaptive or acquired immunity. My immune system is learning. It is learning which microbes are in the environment and getting better at fighting those microbes I'm likely to find. Neutrophils don't learn. Eosinophils don't learn. The acid in your stomach doesn't learn. It does what it does. But we get better at fighting the microbes in our environment as we age because of making these memory cells and these secondary immune responses. Now, this is where vaccines come in place, because if this child was vaccinated, you know, with a fake virus or something which was heat killed or a part of a virus, then they got that primary immune response, which isn't all that good, out of the way with something that doesn't cause infection. And now when they go to school, if they ever encounter that microbe, they are, you know, ready to have this secondary response. Now, let's say this person wasn't vaccinated. So now if they go to school and this, you know, microbe is going around, they are going to react with their primary immune response. And once again, they could get really sick, right? They might not be able to mount an efficient defense before, you know, this gets to be an urgent scenario. In the future, all right, she will then have memory cells and be better at it, but she didn't the first time she met this virus. So that's the purpose of vaccines, to get that primary immune response out of the way, to generate those memory cells. Did I miss one? Forgive me. Yeah, let's skip it. And to get that, oh no, this isn't, okay. And to get that first one out of the way. Now, those plasma cells, they can make thousands of antibodies per second. Once again, thousands of antibodies per second. So this is an incredible factory to make these antibodies. And notice they're being secreted, all right? So the T cells are different. Their receptors are on the cell surface. These are leaving the cell, these antibodies, they leave. And so as a result, they can be in body's fluids, they can be in breast milk, they can be in blood plasma, they can be in nasal secretions, etc. So here you see a B lymphocyte secreting antibodies into blood plasma. This is why this response is called the humoral response. Now, I know when you hear the word humor, you think of funny, and you're looking at this video, and it's not all that funny. So what's up with that? Well, the answer is, here we're using the Greek word humor, which means fluid. The ancient physicians, like, you know, Hippocrates and Galen, you know, they were very worried about the humors of the body, you know, where the humors were in balance, if you know your medical history. Humoral response means this. Let's say that I get a serious disease. Well, I might die. I mean, with a lot of diseases, you have 10% of those who get it die. 20% of those who get it, there were some forms of Ebola where more than 90% of people who got it die. So I could get a disease, and odds are I'm likely to die. All right, even though some people survive, odds are, just playing the statistics, it's not going to be me. However, if you got the disease and you survived, and you give me some of your blood plasma, now I might have a very good chance of surviving. So your plasma can confer immunity to me. Your plasma could save my life. What's in plasma? It's mostly water, and then there's some proteins. These proteins, all right, so if you remember, the plasma includes globulins, like the immunoglobulins, Ig, like IgG. So here, plasma is having these antibodies specifically for this microbe, all right, whether it be Streptococcus, pyogens, or Staphylococcus aureus, or whatever. These are secreted from the body, they are, or the cell, they are now in the humors, the fluids, hence humoral immunity, and hence a humor, a liquid that doesn't have any cells, a cell-free liquid can confer immunity from one person to another. That's why this is called humoral immunity. So these antibodies are really important. What do they do? A lot, all right? And so I'm gonna go through a bunch of ways that antibodies promote immunity and fight microbes. But then there's one, there's maybe two really important ways, one being the most important. We'll end with that. So first off, let's say that this is, I don't know, a virus, a toxin, a something. And the reason it screws up the body and that is dangerous is, let's say it can pass through this channel or bind to this receptor or something. So it fits somewhere that we would prefer that it didn't fit. Because it fits in that space, that's bad. It does bad stuff when it fits in that space. However, once antibodies stick to it, it doesn't fit anymore. All right, so it won't fit in that receptor. It won't pass through that channel. So just by having antibodies stick to stuff, that's good. Now, maybe this toxin is no longer, dangerous. So that's one effect of antibodies. Lots of microbes move. Lots of bacteria have flagella. Well, if you make antibodies against the flagellar proteins, guess what? They can't move anymore. They can't spread throughout the body. So, that has gone a long way to stop the infection. These antibodies are Y-shaped. We'll get to IgM, which has like 5 of these stuck together. But so let's look at these things. Let's say these are dangerous, whatever, toxins, viruses, whatever, and they're floating around. But now each antibody can stick to 2 and kind of clump them together, all right? And so here we are sticking them together. And now, instead of these little molecules, which could say, be dissolved in your blood and float around, where it could encounter receptors, et cetera. Now it's one solid mass, which is going to precipitate or is going to settle out. Maybe it's now too big, to circulate, et cetera. So that's another way that antibodies can function. This second video goes through a few more. Once again, antibodies can just stick to things. Now, here you see the ones with the red base, the IgG. At the very end, I'll go through IgE and contrast how that one's different. So here you see, antibodies can just stick to stuff, and that could, prevent it from going places. IgM, if you remember, is a set of 5 antibodies stuck onto a single base. When I teach, the silly analogy that I use is, let's imagine I'm going to rob a bank with 4 of my friends. Well, if right before we go into the bank, the 5 of us to rob the bank, if you could tie us together, well, we're not going to be very good at robbing the bank if you, push us all together. And in the same way, if each of these 5 antibodies can now stick to something, it agglutinates things. It causes them to clump. It limits their mobility. When we are looking for blood type, so let's say I'm trying to figure out if I'm, blood type A or B or O, I would add antibodies and look for clumping. And if I see clumping, I say, oh, I have antibodies which are clumping the, the A stuff, so there must be an A in my blood type because of the clumping. So agglutination or clumping is one of the ways that this works. This neutrophil can recognize bacteria, and if it does, it can eat them. However, it's not perfect, and if you want to speed this up to make the neutrophils more efficient, you can attach antibodies to the microbes. So while neutrophils might recognize a bacteria on its own, once an antibody sticks to it, they're really good then at recognizing those. And so the process of sticking an antibody to something to promote phagocytosis has a name. It's called opsonization. So neutrophils, macrophages, dendritic cells, that's something that they're very good at. They can eat stuff on their own, but having antibodies stuck to something is kind of like an alarm bell going off, if you like, or if you like the analogy, it's kind of like a handle, which, you know, is easily grabbed. And so therefore, this pink microbe with antibodies attached is much more likely to be recognized and to undergo phagocytosis than a pink microbe that did not have antibodies attacked. And so as we release antibodies, even though this is adaptive immunity, you're now going to make the innate immunity work so much better. Neutrophils and monocytes are going to be so much better because of having those antibodies attached. Okay. Finally, so antibodies do a lot of things. Finally, there's one way, which is, you know, by far the most effective way that antibodies work. If you remember blood clotting, the term that was used was a cascade. It's not a one-step process where you don't have a clot. No, now you have a clot. You know, and this one protein makes the clot. No, it was, you know, factor 1 activates factor 2, activates factor 3, activates factor 4, so a cascade. We have another cascade in our body known as the complement factor cascade, or just the complement cascade. Now, there's actually a couple of these. So this is actually really complicated. We have actually different pathways for different cascades. This is good. And it is going to involve, you know, a number of steps. If you get lost as I quickly go through this, obviously you can go through this video slowly, but the key is this blue part here, all right? Now, when I, you know, teach, I sometimes use the morbid analogy. There's a couple things I would prefer don't happen to me today. I would prefer not to get shot. I would prefer not to be stabbed. These things have something in common. They put holes in me. I don't want more holes in me. I don't want to have, you know, bullet holes or stab wounds. If I do, my good stuff leaks out and bad stuff can leak in, like bacteria and et cetera. I can no longer regulate temperature. So extra holes, they're bad. And not just for me. All living things think the same way, even like bacteria and protists and worms and the like. So if you put extra holes in things, those things die, all right? Because bacteria, if you can put a hole in a bacteria, well, its good stuff leaks out. Its bad stuff leaks in. It can't regulate, you know, ion concentrations. So you put a hole in me, I could die. If you put a hole in a bacterium, the bacterium could die. If you put a hole in an amoeba, the amoeba could die. And that's what this aqua set of proteins is. It's a hole, so we'll get there. So what we want to do is essentially stab these microbes through this cascade to assemble these blue proteins on the cell membrane. So it starts off with these Y-shaped antibodies when they stick. Let's say that this is the bacteria here in red. Now, we need more than one antibody sticking, so that could be 2 separate IgGs, but also remember that IgM travels in a group of 5, so IgM already has more than one. It could do this on its own. So once an antibody sticks to something, that now attracts a complement protein. From where? From the blood. Your liver knows that it's a dangerous world. Your liver is already making complement proteins in your blood, so they're there just waiting to be activated. And so the liver plasma proteins are already there. And so then the antibodies bound will simply then now activate one, which activates the next, which activates the next. Interestingly, some of these break into 2 halves. Notice that 2 of the, some of the halves are staying bound to the membrane. These other ones might actually have a separate function, like promoting inflammation. So complement proteins may break into 2 halves, with each of the 2 halves having a function. And so, this video will go through it in a little more detail. There's actually a couple complement pathways. So, if you were an expert, you might get into like this one, that one, C3, et cetera. But then we do a whole bunch of things, and then we get to these four. So these final four all kind of assemble here. And these four are important because once we get these four, then we can now attract the aqua ones. We attract one aqua one next to another one, next to another one, next to another one. And these last, four in the complement cascade then cause those aqua proteins to assemble. Those aqua proteins make the MAC, the membrane attack complex. And once again, that essentially just put a hole in the microbe. You just stab the microbe. Just like I don't want extra holes in me, then this microbe doesn't want extra holes in that. But we just did that. So now it's good stuff is leaking out, bad stuff is leaking in. It can't regulate ion concentrations. So the microbe is dead. You shot or stabbed the microbe, you've killed it. Now, that was so important. It's worth just, whoops. Yeah, it's here. So let me just go back here. A neutrophil can eat the bacterium that it touches. A monocyte can eat the bacteria that it touches. But look at these plasma cells. See, they are secreting these antibodies from their from themselves, maybe even thousands per second. So an incredible volume. Those antibodies, they go wherever. So if this is blood plasma, it's going all throughout the blood. If this is IgA, it's going all throughout the intestinal juices, through nasal secretions, etc. And now these antibodies can start the reaction where you kill a bacteria just by touching it. So this lymphocyte is killing microbes that it will never touch. See, it's sending the antibodies out in all directions, and the antibodies are doing the killing. So they're, kind of like an automatic weapon, and in essence, it's not the gun that's killing, whatever, it's the bullets coming out of it, which are now going in all directions. This is why this is so incredibly powerful. This one lymphocyte that's, say, stuck in a lymph node is now killing bacteria all over the body that it will never touch. So that's, why this is so much more effective than, say, those neutrophils, et cetera. So that's why it's called the humoral response. This immunity is being spread through the liquids that contain the antibodies, the humors. Now, just a couple of additional points just on humoral immunity. IgG, the ones with the red bases, they can just be secreted into plasma and stick to an antigen. IgE is different. It sticks to mast cells. So if you say, I am going to put IgE as the base of this antibody, you're saying that I want this to stick to mast cells. Mast cells are inflammatory cells. They are full of these granules of inflammation. Now, if the antigen binds the antibody, all right, this will cause the mast cell to release its granules, which will cause inflammation. Now, this is actually a really good mechanism, especially for dealing apparently with worms and other larger parasitic things. These worms, and intestinal worms, these are things which has plagued humanity, that billions of people have worms today. They cause, hundreds of thousands of hundreds of thousands, if not millions, of deaths per year. So worms are an incredible source of mortality, not so much in the United States. And so for that, it's perhaps, nematodes and flukes and tapeworms are kind of foreign to us. This is great because if, an antigen from, say, a worm is binding to a mast cell, we start inflammation, which brings all of the immune players to the area of the worm, which is, a great defense against it. In societies where, hygiene and, et cetera, has now eliminated most of these worms, this response now tends to get hijacked a bit, and it can cause allergies. All right, so allergies are then when we're doing this, not in response to worms, which was, you know, kind of the original goal, but now we're kind of hypersensitive. So now we're allergic to grass pollen, to cat dander, to bee stings, to peanut butter. Maybe in mild allergies, you know, all of these inflammatory things like histamine make your eyes puffy or tear, etc. But this can actually be much more serious. It can even cause death because I have mast cells all over my body. And if they all degranulate, that means this histamine is causing vasodilation over here, over there, over there. Now, vasodilation, you know, a little of it is fine. Like I don't mind getting puffy and red in this area where it's infected, you know, here and bringing immune cells here, you know, that's okay. But if that's happening everywhere over my body, I have 5 liters of blood, but I have 8 liters of space. I don't have enough blood to send it everywhere. And if histamine is released everywhere, telling blood vessels everywhere to dilate, I just don't have enough blood. My blood pressure falls, and now I don't have enough blood pressure to get oxygen to my brain and back. I can go into anaphylactic shock, and I could die. All right, and so allergies, which will, you know, I get to in other topics because, you know, this is a component of asthma. Then, you know, what causes this? Is a good topic. Now, this makes a little more sense after the next video, but the next video talks about T lymphocytes. T lymphocytes, they help to train the immune system, among other things. And one of the things that they help to do, if you have, say, a difference between, say, Th1 T lymphocytes and Th2 lymphocytes, they produce different signals. Based on the signals, this could encourage a B lymphocyte to make IgG, which doesn't cause allergy, or IgE, which does. And so if we have more Th1 cells than Th2 cells, then the Th1 cells are sending different signals, which will tend not to make IgE. So, you know, we could train our B lymphocytes in a way that they're more likely to make IgG, all right? And thus we would not be allergic. So if I'm making predominantly Th1 cells, I'm going to make IgG and have a less likelihood of being allergic. However, in this modern world, we often now push our emphasis towards the Th2 responses, and now I'm more likely to encourage my B lymphocytes to make IgE, and we see that, you know, in many societies, allergies are going up, asthma is going up, hypersensitivities are going up, etc. One last thought is, while antibodies are very good at fighting disorders, there is a difference between what's called active and passive immunity. So this individual, if they encounter a bacteria, they then make memory cells. They make memory B lymphocytes, and then in the next video you'll see that there are memory T lymphocytes as well. So this person, because they directly encountered the microbe, they're making the memory B cells and memory T cells, which will make their response so much better. There are other ways of getting antibodies. IgG can cross the placenta and pass immunity onto a fetus. IgA can be secreted in breast milk and pass immunity onto an infant. But neither of these individuals is encountering the antigen so that they make memory cells. And so therefore, while they are going to get immunity against this infection, they are not more likely to fight future infections because they have not made the memory B cells and memory T cells that this individual made. This individual made their own antibodies. These antibodies came from the mother. So that's a difference between, say, passive immunity, which doesn't make you more likely to be protected in the future, versus active immunity, which does. So our adaptive or acquired immunity gets better with time. We get better at recognizing those microbes which are in our own environment and get better at fighting them. So our immune system learns, all right? So that's why we reach advanced ages. Animals which don't have a learning immune system, they live much shorter times. we split this adaptive immunity into two halves. We split it into a humoral immunity, which is run by the B cells, which make antibodies, and then a cell-mediated immunity. Now, I'm going to mention this again in the next video, because this is so complicated. I mean, I've heard students say, I took this microbiology class, and my teacher went through all this, and after an hour and a half lecture, I left the room and said, I have no idea what I just heard, because there were so many cells. So I have a couple of videos that are a little sillier, but with the idea that, once again, if you hear all of these cells talking with accents, and it's kind of in the frame of a battle being fought, that this might help you remember. So in the immune saga, there's, components of it which have, where I have, the humoral response, but then also I have, the innate immungers, superheroes, where you go through, each one, talking about what they do. And so once again, if you're having trouble with this, because this is, I would argue, the second most complicated part of the body after the brain. In addition to the serious videos, I also have some sillier ones with superheroes or with an animated battle, and hopefully that might help you with some of these topics as well.